Hello, I'm here today to go over our notes on the microscope. So this activity or these notes on this pear deck are probably going to take 2 days or so. So I'm going to just kind of read through in case you were absent on day one or day two. So you can use this as a resource. So if you look here, micro means small. So a microscope is an optical instrument used for viewing very small objects. And as you can see, the evolution of the microscope has changed very much over time. Some are very simple, and then as time progressed with our technologies, they get more and more complex. So I think that's just a cool little intro slide. So this is what we're going to tackle. Again, depending on what day you were absent or on quarantine. The history and the scientists behind the microscopes, the properties of resolution and magnification, a light versus an electron microscope, and then the importance of microscopes will be tackled on day one. And then day two, you're going to learn about the parts and functions of the microscope, how we handle a microscope safely, and then the different types of slides, a preserved slide and a wet mount slide. So let's get started. The invention of the microscope made it possible for people to discover and learn about cells. And the microscope is an instrument that makes small objects, living or non-living, look larger. So this is a quick little video. You can play it if you would like. It's about a 2-minute video.
Welcome to Moo Math and Science. In this video, I'd like to talk.
And it goes through the four major, actually five, groups of scientists that were involved in the discovery of the microscope.
Welcome to Moo Math and Science. In this video, I'd like to talk about the history.
So the first one, so you can watch that on your own. Zacharias Janssen, excuse me. It begins with him, and he developed the first light microscope in the 1590s in Holland. So that's a very wonderful portrait of him, illustration. And that's what actually his microscope looked like. So it kind of looks like more like a telescope than it does a microscope. But he basically used a lens to see things, or what he noticed, things look larger using that tube there. Then, of course, it wasn't until the 1660s that Robert Hooke came along. He, along with the next gentleman, Anton van Leeuwenhoek, were really kind of cutting edge of their time and trying to discover the microscope. So he was one of the first people to observe cells. He was an English scientist and inventor. And if you look down to the bottom right there, you see that's kind of what his microscope looked like. It was a compound microscope, which was one of the best microscopes of his time. Compound means that they use two lenses. Like I said, as a background, as a scientist and inventor, that was kind of his important thing. But the more importantly about Robert Hooke, I always think about Captain Hook, but the more important part about Robert Hooke is that he looked at the cork of an oak tree, or the bark of the cork of the oak tree. So if you know anything about a tree, the outside is the bark, and then right underneath that bark is called cork, or cork cambium, and that they're the living growth of the cells. So as that tree gets larger and larger in circumference, they actually grow it from that cork, or that cork cambium. So he took a sliver of that and then put it under his microscope, and that's the illustration that he found, or that he drew. And he said that those little tiny spaces looked like little tiny rooms, and it kind of reminded him of like a jail cell. So he started calling them cells. And that's meaning of the word cells means small rooms. So I think that's really cool for you to kind of get that reference point. Robert Hooke, yes, he used a microscope in the 1660s, but he more importantly coined that term cell that you hear so much in our 8th grade science classes. Next is Anton van Leeuwenhoek, and this is in the 1670s, and right around the same time of Robert Hooke and his discovery. This guy, he was kind of an inventor, and he kind of a jack of all trades. And you can see there, that's actually what he used. He observed tiny objects with microscopes. He actually took like pond water, and he called them animalcules, right? Because he saw these things moving. He didn't know what they were, but he was able to see them with his simple microscope. That's why I laugh. It's in his spare time, he built simple microscopes. Simple microscopes have one lens, whereas Robert Hooke, he had two lenses. Sometimes Anton van Leeuwenhoek is referred to as the father of microscopy because of all his different types of microscopes that he's developed over his lifespan. And then last but not least, circa 200 years later, we have Ernest Ruska and Max Knoll, and they are the kind of attributed with the first electron microscope. They found a better way to achieve a better resolution than just using light, and instead he used a beam of electrons. And there's actually two different types of electron microscopes, which we'll talk about a little later in the lesson: scanning electron microscope and a transmission electron microscope. So that duo, or that pair, in 1931 discovered that. So this is your knowledge check to kind of match each of the appropriate scientists with what their discovery is. What type of microscope did Van Leeuwenhoek make and use? Quiz yourself. And then lastly, what type of microscope did Ruska and Knoll make and use? All right, so our next topic is going to be on the properties of the microscope. And if you look at, I love this photo, but resolution and magnification. So obviously, if you look at this photo, you can tell that you're zooming in on that nose of that very cute, adorable dog. That's magnification. You're zooming in. You do it all the times with your phones, or if you take photographs all the time, you're zooming in. That's magnification. Whereas resolution is dealing with the clarity of the actual object. A lot of times I like to refer to resolution as, like, if you wear glasses or contacts, if you don't have those glasses or contacts in, your resolution is blurry. It's not clear, right? But when you put those contacts in or you put those glasses on, it becomes clear, right? So when you're using microscopes, your goal is to have a good resolution and magnify the image. So let me first talk about magnification. This is the first property, and it's the ability to make things look larger than they are. The lenses in a light microscope, or a compound light microscope, which is the ones that we're going to use in class, magnify an object by bending the light that passes through them. So that's kind of just a visual. That's not done with a microscope because obviously trees are really large, but just to kind of show you that concept of magnification. A compound microscope uses more than one lens. As a result, it can magnify an object more than one lens by itself. So they're going to be more powerful than just a regular magnifying glass. So the compound microscope, which is almost identical to the ones that we're going to use in class, the first lens is down near the specimen, which is kind of the object in view, and that's where the blue arrow is. They're in your objective lenses. And then the second lens is up near their eye. We call it the eyepiece, and that's where the second lens is. So you have two lenses in your compound light microscopes. You're going to have to magnify or figure out the total magnification of that object. So as a result, the total magnification of the microscope is equal to the magnifications of the two lenses multiplied together. So if you look here, this is the eyepiece. It's a 10x. That x value is typically right after the number. And then in this case, this is your medium, and you also have a 10x. So 10x times 10x equals 100x. So you're looking at that specimen through the microscope, as long as it's clear and magnified, at 100 times its regular vision, okay, with just the naked eye. So these are your examples. Again, microscope 10x times 10x or 400x. So please do this practice problem. And your correct answer is 40. Okay, tell me the correct formula. How do you calculate total magnifications of our microscope? Okay, next is resolution. To create a useful image, a microscope must also help you see individual parts clearly. The ability to clearly distinguish the individual parts of an object is called resolution. So you can see those three objects right there, very blurry. You can't see what it is. Some people say, oh, that looks like Times Square. And it is Times Square, right? You can infer, but it would be a lot better if it was more clear. Okay, so that's kind of your purpose of your resolution. Resolution is another term for the sharpness of an image. And these are kind of two different pictures to kind of show you what I mean by resolution and magnification. So if you look to your left, that's actually an image of magnification. Obviously, you can see some viruses and some bacteria there. And to the right, you can see the blades of grass. And then you can see that little picture. It's very blurry, right? So that's resolution. You need to get it clear so you can see those objects. So what we're going to talk about right now, last part of the lesson, is your electron microscopes. There's two types: the transmission electron microscope and the scanning electron microscopes. As you can see, both are very, very fancy and way expensive, so we don't have them here in the classroom for classroom use. But if you were to go to different museums or laboratories, or even if you go to college, right, in the science room or the science wings, they would have these microscopes available to you. They magnify way more than a regular compound light microscope, just based on they're using a beam of electrons over light. So electron microscopes use a beam of electrons instead of light to produce a magnified image. You can obtain pictures of extremely small objects, much smaller than those with a light microscope. So this first one is called a TEM, or transmission electron microscope. 200,000 times its magnification. Typically, it's a 2D image, typically black and white. The organism must be killed because you shoot that beam of electrons right through the organism to see the inside of an organism. So these are some samples. You can see that's the inside of a cell, and that's the eye of a fly. So you can see the rods and cones coming out of there. So how it actually works, the magnets guide the stream of electrons towards the specimen, and it's projected through a photographic plate. The disadvantage is that the specimen needs to be killed because it needs to be shoot through the electrons through the organism. So that's kind of, you can see how fancy they are in terms of the transmission electron microscope. The scanning electron microscope, instead of creating a 2D image, it creates a 3D image, which is awesome, and it's 100 times its regular magnification. The cool thing about the scanning electron microscope, a lot of times you can see colored pictures because you're just looking at the outside of an organism, or you can use kind of staining purposes to see those images. So these are some images that you can see. Diatoms, sperm on an egg cell, and that's a virus invading a cell. This is another, that's human lice on the scanning electron microscope, what it looks like. So it's not sliced for viewing, but the specimen is sprayed with a fine metal coating, and like the transmission electron microscope, the living cells cannot be viewed because it's going to be on the surface, right? So you're not shooting it; it's not going to die, but it's more on the surface, and it creates that 3D image of those specimens, whatever you're looking at. So, what do electron microscopes use and produce magnified image? Hopefully, that's an easy question for you. What's the benefit of a transmission electron microscope? And then that's a brain pop on microscope. So please feel free, you can kind of see the value of this. You can see this with the naked eye, the light microscope, and of course, it magnifies so much more with the electron microscope. So this is going to be, up until that point, that's day one. This is going to be day two. We're actually going to be doing more application. And we're going to show you the microscopes. We're going to have you, touch and use them in day two. But we have to go over what the parts are. So you see the eyepiece, the tube, sometimes called the body tube, the revolving nose piece, which is here. I kind of think of a revolving door for that one. You have low, medium, and high power. Low is the smallest one, medium is in the center, and then high power is the largest. Stage clips go on your stage. I think of a stage in theater. That's where your slides are going to go. Your diaphragm, that's a tricky one. That's kind of what regulates the amount of light that goes into the specimen, your light source. Your base, your fine and coarse adjustment knobs. Okay, they're right there. And that's everything. Okay. And then here is the functions of all those things. So you can kind of read through what the values or the purposes of them are. Most of them are pretty self-explanatory there. So you always carry the microscope with two hands. You turn on the microscope and place your slide on the stage. You always start with the low power objective lens, which in our case is 4x. And then you'll go to the medium, which is 10x. And then lastly, you'll start, high power is going to be the fine adjustment knob. I usually say low power is 4x, and that's going to be you're going to use your coarse adjustment. Medium is either fine and coarse, so you use both of those knobs. And then lastly, under high power, that's your fine adjustment knob. So you have to be very, very mindful of that when you're using it. Oops, move too quick. Keep the stage dry, and always make sure the bottom of your slide is dry before putting it on the microscope slide, and then put it always to low power before you put it away. This is how to make a wet mount slide. So we're going to be showing you that in class next week. We're going to be working on this, making your own wet mount slide.
Hi there, boys and girls.
And then this is the actual steps, kind of written down for you. Obtain a clean, dry slide. Put your specimen on the center of the slide. Add a drop of water. Gently drop the cover slip into place. The whole cover slip should be in contact with the water. I call it drag and drop. And that's how you create a wet mount slide. Okay, so that's the video lesson for today, or for day one and day two of microscopes. If you have any questions, please feel free to reach out to your teachers. We would love to help. Have a great day.